Thanks for tuning in. I'm Evan Palmer-Young of the USDA Bee Research Lab, discussing our recent preprint on a heated interaction. High temperatures augment inhibition of parasites by a honeybee gut symbiont. The basic question in this project is how temperature influences parasites by modulating interactions between parasites and symbionts. Temperature and the symbiotic microbiota are two factors that are known to affect the host resistance to infection. Common response to infection in humans and other animals is fever. That's an elevation of body temperature. These high temperatures can directly inhibit parasite growth, and they can also potentiate host immunity and ameliorate infection, especially with heat-sensitive parasites. This can happen in mammals, reptiles, insects, and even plants. The symbiotic gut microbiota can also improve resistance to infection, acting as physical and chemical barriers to parasite establishment. If temperature and the microbiota can each affect infection on their own, how does temperature affect the balance between the symbiotic and parasitic microbes and change the ability of symbionts to counteract establishment by parasites? Answering this question is key to understanding how climate and host temperature regulation, including the constitutively high body temperatures of endotherms, warm-blooded organisms, and the infection-associated elevations in body temperature known as fever, could each affect parasitism via symbiont-driven effects. Honeybees and their associated microbiota offer a great opportunity to study the joint effects of temperature and gut symbionts on parasites. Honeybee colonies have an endothermic temperature regulation that's quite rare among insects. They can keep the colony at close to mammalian body temperatures for long periods of time. This is much hotter than what's typical of most insects, but like other insects, honeybees are subject to wide temperature variations during outdoor foraging and in the winter. Honeybees also have a core gut microbiome that's dominated by bacteria from just 4 genera, including lactobacillus species, that are culturable outside the host. These bacteria are acquired via social interactions in the colony and can enhance resistance to gut infections. One group of parasites that's likely to be directly affected by the gut microbial community are the hindgut-dwelling trypanosomatids. These parasites include Crithidium malefici, discovered in Australia in the 1960s, and the currently more common Lotmaria possum, named right here at the Bee Research Lab. Trypanosomatids have been found in over 80% of colonies in some national surveys, and associated with colony collapse on 3 continents, as well as shortening bee lifespan in some experiments. These parasites are phylogenetic relatives of the insect-vectored neglected tropical diseases that cause leishmaniasis. Leishmania infection in the insect stage is influenced by the microbiome of the sandflies that transmit this infection. The honeybee parasites are even more closely related to Crithidia bombi, a trypanosomatid of bumblebees, whose infection is strongly reduced by gut microbiota, including lactobacillus species closely related to those found in honeybees. Our previous work showed that the effects of these bumblebee symbionts on parasites is magnified by high temperatures. This graph shows that the lactobacillus symbiont had minimal effects on parasites at cooler 27-degree temperatures, but left parasites pickled and unable to swim at 32 degrees and above. Here, we addressed how temperature changes the effects of honeybee lactobacillus on their parasites, asking, first, how do symbionts and parasites differ in responses to temperature? Second, do symbionts affect parasite growth? And how does temperature alter the symbiont's parasite inhibiting effects? First, we measured how temperature affected growth rates of symbionts and parasites across a temperature range that might occur in bees. We found that symbionts were more temperature responsive and heat tolerant than were parasites. So growth rates, shown on this y-axis, increased more rapidly with temperature, shown on the x-axis, in the symbionts, changing by around 7-fold over a 15-degree temperature range. In contrast, rates changed by less than 2-fold over a similar temperature range in parasites. You can see these curves are not as steeply sloped. Growth rates also peaked at higher temperatures in the symbionts relative to the parasites, up around 40 degrees Celsius versus 35 degrees or so in the parasites. This graph shows some of the traits estimated from the curves on the previous slide. The symbionts had greater responsiveness of growth to temperature, as indicated by their more steeply sloped thermal performance curves in that previous graph, and higher temperatures of peak growth rate, especially in comparison to the more common trypanosomatid, Lotmaria, which had a peak growth rate some 6 degrees Celsius lower than that of the lactobacilli. As a result of these different responses to temperature, the growth rates of symbionts relative to parasites increased strongly over a temperature range found in bee colonies. The change in these relative growth rates was especially strong within the temperature range that bees use to incubate their larvae and their pupae at the center of the colony. The ratio of lactobacillus rates relative to Lotmaria changed by over 3-fold over just a 4-degree temperature range. Symbionts also inhibited growth of parasites by a production of acids. The acidic spent medium in which these two strains of lactobacillus had been growing fully inhibited growth, shown on this y-axis, of both Crithidia and Lotmaria. This was the case for spent media from both of the two lactobacillus strains, shown in these two panels. However, neutralization of that spent medium back to the pH of fresh media restored parasite growth almost completely. And that was the case for both of the trypanosomatids, illustrating that acidity of the spent medium was necessary for parasite inhibition. Conversely, acidification of the fresh media, shown in this right panel, to the pH of the spent media reproduced the inhibitory effects of that spent media, showing that acidity alone was sufficient to reduce parasite growth. Next, to see whether these inhibitory effects were altered by temperature, we measured growth of parasites with and without symbionts across a temperature range that might be experienced by honeybees. This graph shows that the presence of lactobacillus, indicated by this red shaded curve, had proportionally stronger effects on parasite growth, shown on this y-axis, as temperature along the x-axis here increased. So whereas symbionts had little effect at low temperatures, they severely limited the ability of parasites to grow at high temperatures, knocking back growth by over 50% above 33 degrees. Comparing growth rate of parasites in the presence and absence of lactobacillus, the presence of symbionts reduced the peak growth rate of parasites by almost 50%, shown on the left here. And this is all for Lotmaria. We pursued these findings with Lotmaria rather than Clythidia because the Lotmaria is much more common currently. The symbionts also reduce the temperatures needed to inhibit parasite growth rate to half of its peak value, a measure of the parasite heat tolerance shown in this right panel. Now, although this change was less than 2 degrees, that difference could be quite biologically significant, since the presence of this lactobacillus could then essentially preclude the ability of parasites to grow at these 34 to 35 degree temperatures experienced by newly emerged bees. Whereas in the absence of symbionts, parasites might be able to handle those temperatures. The reason for this reduction in the parasite heat tolerance is that the effects of the symbiont are not uniform, but they increase and strongly with temperature. This graph shows that the symbionts have much stronger inhibitory effects shown on this y-axis, as the temperature on this x-axis gets past around 30 degrees. Now, the temperature range of the center of the colony during the growing season is usually around 34 degrees, which would seem quite favorable for the symbiont-mediated parasite inhibition, whereas the lower temperatures that are experienced at the colony periphery during the winter and outdoors appear to weaken these inhibitory effects. Recall that the inhibitory effects of the symbionts was due to their production of acids a couple slides back. Symbionts produce acids more rapidly at high temperatures, lowering the pH of the growth environment. And these acidic conditions were associated with stronger inhibition of parasites. The pH range recorded in the bee rectum ranges from around 4.2 to around 6. So our results suggest that a pH, shown on this x-axis, on the lower end of that range, that is more acidic pH, would strongly inhibit the growth of Lotmaria. So to illustrate these findings, high temperatures can directly inhibit growth of parasites, but it can also accelerate the growth and the acid production of symbionts, thereby magnifying the symbionts' parasite inhibiting effects. A quick recap. These experiments show how temperature can potentiate the protective effects of gut bacteria against parasites. The core honeybee symbionts had greater tolerance of high temperatures than did parasites, and the symbionts inhibited parasite growth via production of acids, with greater growth rates, production of acids, and inhibitory effects at high temperature. As a result, the symbionts limited the ability of parasites to grow at high temperatures that were tolerable when the symbiont was absent. For bees, our findings predict that socially enabled maintenance of high colony temperatures could promote the establishment of the non-pathogenic core gut bacteria and their inhibition of parasites. Conversely, bees that are unable to achieve these high temperatures may not realize the full benefit of their symbiont community, and bees lacking gut symbionts may not gain all the potential benefits of maintaining high body temperatures. More broadly, these experiments use honeybees, which are facultative endotherms, and their gut microbes as models to help better understand the value of endothermy as a regulator of the microbiome, a modulator of symbiont function, and possibly a symbiont-mediated defense against parasites. Our results suggest that in hosts that have heat-tolerant symbionts, high body temperatures could reduce infection by harnessing that antiparasitic potential of the gut microbiome. You made it to the end. You earned your poetic summary, a B-grade limerick. Bee bacteria thrive in niche warm, giving parasites cause for alarm. When stewed in their acid, invaders grow flaccid. When heated, they ooze sour charm. With that, I'd like to thank my co-authors, colleagues, family, and sponsors, and thank you for your audience. Please get in touch by email with questions, and check out our preprint on bioRxiv for all the details. I'll leave a link in the description.